So welcome, everyone. We're pleased that you could join us for our Living with SADS series. Tonight's topic is Gene Therapy Part 2. I am Marcia Baker, the Program Director for the SADS Foundation. So I'm glad you decided to join us for an hour for our webinar here. A couple reminders before we start. One, please keep your audio muted during the presentation to improve the sound quality for everyone. Your video camera is optional. Following the presentation, we've allotted time for your questions. You may type your question in the chat anytime, and we will address them after the presentation. And now it's my pleasure to introduce our presenters. We welcome back Dr. Andrew Landstrom, who spoke for our first gene therapy webinar last month. If you missed it, it was recorded and is available on stopsads.org website. So Dr. Landstrom is a pediatric electrophysiologist at Duke University who specializes in genetic arrhythmias. He's also a physician scientist and cross-trained in human genetics and molecular physiology. He has an active research program that brings together clinical medicine, functional genomics, precision medicine, and molecular and cellular biology. Joining him tonight will be Dr. Aravind Asokan. He's the director of gene therapy at Duke University. He is a professor of surgery, biomedical engineering, molecular genetics, and microbiology. His research interests include viral engineering, RNA engineering, gene editing, regulation, and regenerative medicine. His research lab studies the biology of adeno-associated viruses and uses cutting-edge molecular evolution to discover new synthetic viral strains. Their goal in his lab is to build transformative technologies to enable the clinical translation of genetic medicines. We're honored that both Drs. Landstrom and Asokan are volunteering their expertise and time with us tonight. So thank you both on behalf of the SADS Foundation and our families affected by genetic arrhythmias. I also want to thank our industry partners who have provided an educational grant to help make this webinar possible. So thank you to our partners, Stride Bio and Medtronic. So now I'll turn it over to Dr. Landstrom to get us started.
Thank you, Marcia, and thanks for the invitation to come back. Thank you all for joining us. Aravind and I are absolutely thrilled to be here to share a little bit more about advances in gene therapy, specifically about arrhythmic cardiomyopathy, which was a hot topic on our last webinar together. So I have no disclosures or conflicts of interest, although I do accept research and salary support from the following institutions. So the questions that we're going to seek to answer in our hour together is we're going to briefly recap what is gene therapy. We're going to talk about what is arrhythmic cardiomyopathy. And this is kind of a newer name for a disease that's been around for a while. You may have heard of it as arrhythmic right ventricular dysplasia or arrhythmic right ventricular cardiomyopathy. We'll just refer to it as arrhythmic cardiomyopathy for simplicity. We'll talk about why AC might be amenable to gene therapy. And is gene therapy the only avenue being pursued for new therapies? And then we'll get an expert's perspective on what are the major challenges for gene therapy and how can we overcome these. And that'll be Dr. Asokan. And then we'll close out with a few thoughts on how patients and families can stay informed and maybe even get involved. So you might remember our time together last time was talking a lot about cardiac channelopathies and how cardiac channelopathies are amenable to gene therapy. And we talked a lot about the different kinds of channelopathies like long QT syndrome, where the defect there is actually in the heart resetting electrically. Short QT syndrome, which is the exact opposite of long QT syndrome, where the heart resets electrically too fast. Brugada syndrome, where the electrical defect there is a loss of sodium current in the heart, which leads to an arrhythmia predisposition. CPVT, or catecholaminergic polymorphic ventricular tachycardia, which is a long word, but basically it means that during high adrenergic states, high adrenaline states, the heart can go into an arrhythmia. And then when we know there's an arrhythmia but we don't know the cause, we call that idiopathic ventricular fibrillation. Last time we were together, we talked about all of these things. Now all of these are potentially causes of sudden death in children, young adults, and older adults. And all of these are genetic, and thus all of them may be amenable potentially to gene therapy. Just to briefly recap what we talked about to kind of ground ourselves for tonight's discussion, we talked about all of these diseases being genetic in cause. And if you took cells from an individual, like cheek cells or blood cells, and you looked in the DNA and you found one misspelling in the right area of the genome, this is a sequencing graph. These peaks are all the different letters of the human genome, and then you can see right here, there's actually two peaks where there should be one because there is a misspelling on one copy of the genome. That is enough to lead, if it's in the right place at the right location, to this. And so this is actually an ion channel that we can study in the lab. And this single misspelling out of all the billions of letters in the human genome, if it falls in the right place, in this case, it fell into an ion channel that's expressed in the heart. And that ion channel is the cause of long QT type 1. And if you look here to the left, you can see these big lines that stretch out from the base. They go up very high in the graph. That's because this particular ion channel is moving lots and lots of current. With this misspelling, those lines flatten. And so one misspelling in the right area of the genome can lead to a complete loss of function of this protein. And this is enough to cause long QT type 1. And so if you think back, if you think back to the basic mechanisms of cardiovascular genetic disease, the whole reason that we can approach gene therapy in this space is that these misspellings in the genome cause a misspelling in the RNA, which causes a misspelling in a protein. And when that protein folds, sometimes it isn't made, sometimes it goes to the wrong place, and sometimes it gets to the right place in the cell, but is dysfunctional. And that single misspelling that's in the gene is carried all the way through to that protein and is sufficient to cause disease. We call these single nucleotide variants or missense variants. You might remember that sometimes we can have a mutation or a spelling error in the DNA that's so severe, it's not one letter, but the entire message gets ended early. And so the RNA gets ended, the protein is never made, and we call this a loss of function. And so either of these are possible. Either of these things can cause different types of channelopathies. And it's certainly the case too with arrhythmic cardiomyopathy that you can have these kinds of mutations. But the idea is that since the mutation is in the human DNA that you and I all have, that it's amenable to gene therapy. If we think back to that definition of gene therapy, I like this one the best. Perhaps Dr. Asokan's got a more refined one, but for simple guys like me, I think of gene therapy as any genetic modification of a cell that produces a therapeutic effect. And whether you're actually changing that misspelling in the DNA, or you're doing something else to augment that loss of the DNA, that's gene therapy, if you're using DNA to do it. And, because all of these channelopathies and cardiomyopathies, all of these things that can lead to arrhythmic sudden death are genetic, then gene therapy holds tremendous promise in treating cardiac ion channelopathies. And we talked a lot about these in our last webinar. To briefly kind of recap, you might remember a discussion about Brugada syndrome. And so if anyone is familiar with this diagnosis, the misspelling here is in the gene SCN5A. That gene makes the cardiac sodium channel. And we talked about those loss of function mutations. Well, in Brugada syndrome, there's a loss of function mutation in SCN5A, which means only half of the sodium channel is made. And you need 2 copies of that sodium channel to have a normal heart. And so there has been work done, and this is work done by one of my colleagues here at Duke, who has basically found a tiny little bacterial ion channel that can be re-expressed in a heart cell and potentially make up for the loss of the genetic loss of function mutation. And this is work done by Ninad Bursak here at Duke. So an interesting potential gene therapy tool for Brugada syndrome that's coming down the pipe. We also talked about long QT syndrome. Long QT syndrome a little bit different. There's lots of different kinds of long QT syndrome. We talked a little bit about long QT syndrome type 1, how that's caused by misspellings in a gene that causes a dysfunctional protein to be made. And it's not sufficient to just remove that dysfunctional protein. You actually have to replace it. That way you have 2 copies of the normal protein. And so we talked about work by Dr. Ackerman, where he has shown, at least in cells, that you can silence, you can quiet down that mutant KCNQ1 gene, that spelling error, and actually then replace the normal KCNQ1, and that cell is actually cured of its long QT. Now, with both of these approaches, we have a long way to go, of course. These experiments are done in cells, and while they're very exciting, we still have lots of road ahead of us to be able to apply this to humans, and we'll hear a little bit more about that from Dr. Asokan. Well, let's pivot now, after that kind of reminder of what we talked about last time, and talk a little bit more about arrhythmic cardiomyopathy. And so you might remember last time there was a great question about what are we doing for arrhythmic cardiomyopathy? What's in the field? Well, arrhythmic cardiomyopathy, I think, is actually an excellent candidate to consider for gene therapy. And for those of you who may not know, arrhythmic cardiomyopathy is a very distinct disease. What makes arrhythmic cardiomyopathy is what happens at the tissue level. And so if you look at a heart of a patient with arrhythmic cardiomyopathy, the muscle of their heart is actually replaced with scar and with fat, okay? And you can imagine that that's not good for something that needs to be a well-formed, efficient muscle. The scar and that fat progresses, and it can lead to two things. It can lead to failure of the heart to be able to squeeze well, to pump blood out to the body. We call that heart failure. And this can happen in isolation or together. They can get ventricular arrhythmias. So what that means is that this tissue in the heart is actually disrupting the electricity in the heart, allowing for misbeats of the heart to occur. If it happens in the bottom chambers of the heart and they go on for a prolonged period of time, these can cause fainting or maybe even be life-threatening. And so arrhythmic cardiomyopathy has its name because it's a heart muscle disease that can lead to heart failure or arrhythmias, arrhythmic cardiomyopathy. AC is a big problem. You may not have heard of it, but it's actually more common than you think. You may have heard of cystic fibrosis, right, where kids get these very thick secretions in their lungs and sometimes need lung transplants. Well, arrhythmic cardiomyopathy, AC, is actually 2 times more common than cystic fibrosis, just to give you an idea of how common it is. Still a rare disease, but more common than you might think. It has a very high mortality, so left untreated, it's as high as 2 to 4% per year. That means if you have AC, that's the risk of dying per year. And we have medications that can help, but nothing that can cure the disease. And then there's variable manifestations of disease. So arrhythmic cardiomyopathy, much like all the other channelopathies you talked about, are genetic. It's a genetic disease. And even in families where we know the same spelling error has occurred in multiple family members, there can be variable ways that spelling error manifests in the family members. Some people may have no disease at all. Some people may have that spelling error but have pure arrhythmias. So it's really hard to predict how a spelling error in a family is going to progress in each patient who has it. And, despite how terrible this disease can be for some, there are no medicines that we have to prevent or reverse disease. We can do things like suppress the arrhythmias with things like beta blockers, but we can't actually fix the disease, and the disease is progressive. To make matters worse, it's known that exercise, particularly strenuous exercise, actually worsens the disease. And so people who are at risk of developing AC or who have AC are often told to be restricted from sports. And that can be really challenging for some. And also, if we know that somebody who has a healthy heart is given a beta blocker and told to sit on the couch for the rest of their life, we know they're going to get heart disease too. So it's really a delicate balance of how we actually do this. And it's challenging because we don't have great medicines to prevent the disease from progressing. And so this is where I think potentially gene therapy has a role. So what causes arrhythmic cardiomyopathy? Where are those misspellings that actually cause the disease? Well, if you think about a heart, shown here to the left, and that heart is made up of heart cells, shown here to the top right. Those heart cells have these little rivets that sit between the ends of the cells. We call these desmosomes. And I think of these rivets almost like the rivets on a jean, like on the pants that you may have on right now. And it's really important because those rivets hold the ends of the cells together so that when your cardiac myocytes contract, they can pull on each other, transmit that force across all the different cells of your heart, and lead to contraction of the myocardium. So without these, it's almost like you put your hand in the pocket of your jeans and your pocket opens up. And then you do it again, your pocket opens up even more. Before you know it, your pocket's pulled off of your jeans. Same thing with arrhythmic cardiomyopathy. Misspellings in the cardiac desmosome disrupt this rivet and actually pull the cells apart with every heartbeat. And this is one thought why exercise and having high heart rates for a prolonged period of time serves to worsen the disease progression. So arrhythmic cardiomyopathy is caused by genetic mutations that disrupt the rivets, the desmosomes, that are between heart cells. And if you zoom into these rivets at the molecular level, there's lots of proteins that go into it that make up these rivets between the heart cells. And they have kind of long words or long names like desmogliin, desmocolin, desmoplakin, plakoglobin, plakophilin. These are some of those proteins, just sort of shown diagrammatically how they fit together. Mutations in every single one of these proteins can cause arrhythmic cardiomyopathy. All right, so you may have arrhythmic cardiomyopathy due to a mutation in PKP2, plakophilin, or DSG2, desmogliin. And we know some people have mutations in more common genes than others, but PKP2 is the most common genetic cause of arrhythmic cardiomyopathy, with about 30% of all patients who have AC having mutations specifically in PKP2. And PKP2 is very specific because it's a loss of function mutation. So the mutations that people have lead to loss of one copy of PKP2, and your cells need two copies to be able to make those rivets. And so without that, the rivets actually tear apart. And so there's been a lot of thought that's gone into, well, potentially, could this be something where you could intervene to fix the disease? Right? And so with that kind of introduction, I'll turn things over to my colleague, Aravind Asokan, to talk to you a little bit more about where we are on the cutting edge of gene therapy for arrhythmic cardiomyopathy. Aravind?
Thanks, Andrew. Thanks, everyone, for inviting me to participate. Can you guys hear me okay? Yep, thanks. I so my background, I'm an organic chemist by training, and somewhere along the way, I turned into a virologist. I was at UNC Chapel Hill, so I still bleed light blue. In 2018, I moved to the dark side, being on the Duke faculty for the past 4 years, where I have had the good fortune of meeting Andrew and understanding some of the awesome things he's doing in his lab to understand the mechanistic underpinnings of cardiac disease. My lab, everybody that comes in, they're technology junkies and they're tinkerers. And we tend to look at biological macromolecules, whether it's proteins or RNA or DNA, that nature's been so elegant in designing. And we think about how we can redesign these tools and use them in the context of medicines that can be transformative. The crew you see here, they do all the work. As Andrew pointed out, we're just here for the sales pitch and the 10,000 bird's eye view perspective that we can provide. Some conflicts, had the good fortune of founding some companies along the way based on these platforms, and acknowledgements to collaborators and funding, without which this won't happen. But let me dive into sort of how we got into the gene therapy space and maybe give you a little bit of background of these viruses that are being used in the field currently. So next slide, Andrew. I should point out before I dive in, the American Society for Gene and Cell Therapy maintains a really up-to-date clinical trials and gene therapy 101 database. That is a fantastic resource with a lot of input from a lot of thought leaders in the space. And it's very up to date on the data. So I'd strongly recommend checking on this resource whenever you have time. It's super useful to browse all the activity in the space. Next slide. So when we start thinking about gene therapy in the context of any indication, whether cardiac or muscle or liver or brain or ocular, there are a lot of considerations that go into, well, how are we going to design this therapeutic? And it's intricately tied in all the way from the patients that we think about to the genetic basis of the disease. Some of the points which Andrew touched on are these dividing cells that are going to rapidly divide and lose the genetic material that we're providing to them, or are they non-dividing, in which case we're able to target them effectively? Is it a protein that's secreted into the blood, or is it something that needs to stay in the cell? Or in the case of PKP2, does it have to do with the infrastructure, the architecture of the cell itself? Do we need really high levels at a specific spot, or is it broad expression? Do we need it in a tissue specific manner? Can we turn it on and off? How do we get the genetic material there? Which kind of virus makes the most sense to use? Or do we use a non-viral platform like polymers or other reagents that can deliver genetic material? And does the genetic material have to be DNA? Why can't it be RNA? And if so, what are the tweaks there that we can make? The routes of injection, should we be immunosuppressing? Can we redose afterwards? What does it mean from an insurance perspective? These are all the broad questions that the field grapples with. And we start to look at these as sometimes one parameter at a time, but a lot of these do meld together very effectively in the way we go about doing some of these strategies. So with this backdrop in mind, if we can filter down to, well, how are we going to get there? What is gene therapy in the context of delivery? I'll talk to you about this virus that we started working on that can enable this. Next slide. So there are a number of different viruses in nature that scientists have figured out over the past decades that they can actually hijack what they do, which is package genetic material and then infect cells to deliver the genetic material. But you can really short circuit how these viruses package the genetic material and get them to package DNA or an RNA of interest that's coding for a therapeutic product. For instance, PKP2, or it's a secreted protein in hemophilia, it's factor 9 or factor 8, and so on and so forth. And many viruses can do this. And the one on the left is called adeno-associated virus. It's a really tiny virus, about 25 nanometers. The one next to that is the adenovirus, which I'll tell you about the relationship between these two in a second. That one's bigger. It's 100 nanometers. There are respiratory infections, and some of the GI infections that we get are typically from adeno. And then there's lentivirus. HIV is the most well-known lentivirus, and it has been repurposed beautifully by researchers to replace the genome. But lentivirus only goes to certain types of cells, adenovirus like certain other types of cells, and then AAV is this whole spectrum that I'll touch on in a second. Next slide. So this is an interesting story. So you can see it's 1965, and it was one of the first papers that came out. And scientists were able to see by cryo-electron microscopy these giant viruses on the left. But in the middle of these viruses, they would see these tiny little spherical viruses in the middle. And at first, they thought it was just a defective particle which was assembling. And so it's just a useless part of the other virus. The other virus happens to be adenovirus, which again, as I told you, causes respiratory infections. I mean, our kids, and by the time we were there, age 7 or so, we've seen plenty of it in daycare. And essentially all of us have seen adenovirus at some point in time. But what is this other tiny thing? And it turns out that they were able to purify that and amplify that. And this virus is actually an opportunistic co-infecting virus that only replicates when adenovirus is present. And the virus by itself is non-pathogenic, and it doesn't cause any disease, and it can't replicate on its own. So it's actually a defective virus, and it belongs to the parvovirus family. And hence the name adeno-associated virus is how this whole, and it has a completely different genome, has nothing that it borrowed from the adenovirus, but it's just this opportunistic viral virus that is there. And in many species, I mean, we're talking humans, and you can go all the way from there to bats, to snake, to non-human primates, to mice and sea lions and dolphins, all ubiquitous everywhere. Of course, each one's got a different tweak to it on the surface, but they're all adeno-associated viruses. Next slide. So just some quick high-level points. It's a defective parvovirus, parvo meaning tiny, and it's only 25 nanometers in size. The genome is a DNA genome, and it has 4,700 bases of space that it can package. So not a whole lot of genetic information, but surprisingly a lot of genetic information in the context of the number of genes that have been explored as being genetic material that should be packaged into this virus. As I said, it's non-pathogenic. The immunogenicity is really low for the wild type virus, and it does not replicate when there is no helper virus present. So you can strip the genome, put in your therapeutic gene of interest, and essentially you have a nanoparticle with a viral shell and your genome of choice on the inside. And that's the basic architecture of this virus. Next slide. This was one of the earliest studies that totally got me hooked on this space. Again, looking at it with a chemist's eyes. So this is what the 3D structure of the virus is. It's not, I'm pretty sure it's not actually colored, although it would be awesome if it was red color this way. But I think essentially looking at this and then breaking it down. So Andrew, if you just kind of click through the animation. So it's made of 60 copies of that protein. And the protein comes together as a trimer or a pentamer, and it makes this beautiful icosahedron structure. And it packages genomes. It's got these pores at the pentamer that you can see there on the top right. And the pore is how the DNA gets in and gets out of this virus. So it's figured all of this out just by putting 60 copies of the repeating copies of a protein together. And all these spikes that you see on the surface are how it binds receptors and gets into cells. Once it gets to the nucleus, it uncoats and it releases the genome. And from that point on, you've delivered the genetic material that you have. So this is one of the earliest studies when the crystal structure was solved 20 years ago that really got me started looking into this space. Next slide. So today, how we make adeno-associated viruses as a recombinant virus is there are these different plasmids, DNA plasmids that you can make, and you throw them all into cells, into these producer cells, and the cells just start spitting out your virus of interest. What is thought, and nobody has really been able to solve this over the past 40 years, is these proteins come together and they make these empty shells. And then there's actually a helicase. It's an enzyme that grabs the DNA and then twists it and puts it through the pore and packages it into this virus. And it's this very elegant mechanism that people have tried to study, but you can't really capture it in real time. So there's all kinds of biochemistry type studies that have been done to understand it. But long story short, that DNA that you see, there are tiny hairpins on either end of the DNA that you see there. And all you need are those tiny hairpins on the end. Everything in between those tiny hairpins can be any genetic material of choice. And essentially the virus recognizes those tiny hairpins and then connects it to the empty shell and then packages it all into the empty shell. So you think of it as a conveyor belt with empty shells just going, and then a somewhat efficient process trying to put genomes in as fast as they can. So you end up making some empty shells and you end up making ones with DNA in them, and all of it can be purified, and there's a whole process afterwards. Next slide. So just to kind of cut to the chase here in terms of what an AAV gene therapy product would sort of look like, as you might be aware, there are two approved AAV gene therapy products. One is Luxturna, the other one is Zolgensma in the United States. Luxturna is an AAV that packages the RPE65 gene, which is involved in congenital blindness. And Roche owns that product. And essentially, that virus is injected subretinally into the back of the eye and delivers DNA, a correct copy of the RPE65 gene, restoring vision in those patients. Zolgensma is for spinal muscular atrophy. It's an AAV that packages a copy of the SMN gene, which is the one that's mutated in spinal muscular atrophy. That one's delivered intravenously in children, and it goes to every tissue as much as it can in patients. delivering that copy of the gene, which is missing in those patients. The table on the bottom left is really just for you to give you an idea of sort of how scale has been working in this world. We talk in exponential numbers in this space. It's not in milligram quantities or gram quantities like a drug, but it's more in terms of, well, if you have, how much is 1E14 is 100 trillion particles is how much we can make from 10 liters of a production run. And if you were going into the eye, you could do 10 patients with that dose. But if you were going to do that in a child, you would only be able to inject one systemic dose with that because it's on a per kilo basis and you're trying to get to multiple tissues. Now, if I were to grow that bioreactor to 1,000 liter or 2,000 liter, which again, the field, they've really grown in leaps and bounds over the past 5 years with this. There are multiple 2,000 and 3,000 liter reactors that viruses are produced from. So for example, Pfizer's or Sarepta's muscular dystrophy trials, there the yields are about E16 from a single run. And E16 in an adult still gives you only one potential patient, but if you're going younger, you could get up to 10 patients. So this is sort of how the numbers are in the space. The picture on the right is sort of a miniature lab adapted version of what's really happening. This is a 50 liter reactor in my lab at Duke that we're making viruses with. And you can see that this is sort of, this slush is where we end up making the virus. And then there are a number of steps downstream where you can purify the viral particles with the genomes in them. And that becomes your drug product that you can use. Of course, this is only used in the context of our mouse model studies and any other animal model studies that we evaluate in the lab. Next slide. So one of the cool things about AAVs as we continue to dig into them as a community is nature, as I told you, has made a lot of these AAVs. There are different serotypes, different strains all over the place. And each of them, there are groups of these, of course, but they all tend to have very unique tropisms for different tissues after intravenous dosing. So in this study done by multiple groups that just collated all the data together, essentially what the groups did was package the beetle luciferase gene. And if you inject beetle luciferin into these mice, they light up like a Christmas tree, and you can use a CCD camera to capture that image. And that's what we're trying to do. We're just using the luciferase gene from beetles as a surrogate for DNA that is being delivered by this virus. So you can see, for example, in the first 4 viruses, they tend to go to the liver. You can see the liver lighting up quite effectively, or first 5 even. But when you start going to type 6 or type 8 or type 9, you start seeing this broader distribution. Cardiac tissue lights up. You can see muscle lighting up, and you can see intensity increasing because these viruses deliver the DNA much better. So there's a lot of diversity in nature, and correspondingly, there's sequences that change in the protein, the structures, the spikes, they all change. The receptors that they bind to changes. And so there's a lot of unique diversity here that can be tapped into when we're building these as reagents for delivering DNA. And that's one of the basis for how my lab approaches these problems. So we look at these viruses, we try to understand why they're doing what they're doing. And once you get the molecular understanding of how they do it, you go ask, can you change them the way nature would to specifically go to the heart or specifically infect skeletal muscle, for example? And can they deliver DNA to these specific tissues? And then matchmake with folks such as Andrew, where we can have a meaningful cargo being delivered to the cells of interest. And that's sort of how we approach these problems. Next slide. This all sounds wonderful. There's a lot of colorful viruses and structures and pictures that I've shown you guys, but the field, as we have grown through the field with clinical trials and approved products, there have been lots of clinical challenges that have been posed. And some of the questions that we ask in the lab are very centered around how do you tailor this platform to address these challenges that are coming back to us in a feedback loop from the clinic. For example, how do we get them to specific tissues? If I'm going in with an intravenous dose, that becomes a bigger challenge. How how are we going to address that? As I mentioned to you, a lot of us by the ages of 7 or 10 have been infected by adenovirus, and AAV is an opportunistic infection. So we have immunity to AAV as well. So a lot of us have antibodies against AAV. So patients have to be screened, because if there are antibodies, it'll just shut down the virus, or in this case, the drug product itself. AAVs that work in a mouse may not work in a monkey, but we don't know whether it'll work in the clinic. Some work well in the clinic, but they don't do well so much in the mouse. So there are these disparities, and it kind of makes sense. It's a virus, doesn't necessarily translate between species that easily. So that's a challenge. And there are other questions around durability and toxicity. If you go too high in dose, then these are questions that pop up as well. I'll touch on a couple of these really quickly, but the idea here is, can you leverage this platform to address these challenges and continue to improve the space from the perspective of delivering DNA? Next slide. This is an example of a virus that we engineered that was actually injected intravenously into non-human primates. And on the left there, you see pictures of this monkey that was injected intravenously. And after the final time points and terminal time points, when you harvest the different tissues, which is in the middle panel, you can see expression in the ribs, the quadriceps, the abdominal muscle, the diaphragm, and the heart, but there is no expression in the lung or the liver. So we actually engineered this virus to avoid all other tissues except muscle tissue. And this virus infects muscle, skeletal muscle, and cardiac only after an IV. Now, what is the benefit of something like that? If it is a muscle target, or in this, as Andrew pointed out, if it's in the case of ARVC, there is no reason to express the ARVC copy of the gene in the liver or the lung or in the kidney, where these organs are not technically affected by the lack of that gene. And so here, and if you do, then there is a risk of toxicity that we might get based off of off-target effects. So how do you think about expressing these genes in only organs of interest? And this is one example where we were looking at musculoskeletal disorders in general. And we were able to engineer this virus that can infect heart and skeletal muscle. Next slide. The other challenge, next slide, Andrew, is the impact of neutralizing antibodies. Mavis McKenna was a virologist, structural virologist. She actually passed away from ALS last year. She solved these structures of antibodies bound to the virus surface. All the gray globules are antibodies, and the viruses are the colored center of these structures that you see. The reason why she did this was she wanted to understand why people have antibodies that are neutralizing these viruses. If you understand the structure and if you peel off those antibodies, I can tell what are the regions that the antibodies are binding and neutralizing this virus. So then what my lab did, we came in and engineered these regions so antibodies can't see these viruses anymore. So it's sort of the opposite problem to the vaccine world. We want to induce antibodies to block viruses, but in case of AAV gene therapy, we want to avoid the antibodies so that you can get effective delivery. So we've been slowly engineering the shell to avoid some of these antibodies. And that's led to some very interesting new reagents that we can use. And I've just captured that in a schematic on this last slide, where if you go to the next slide, Andrew. So for example, I'm looking at the protein on the top left. You see the blue spheres and the red spheres. Those are regions that antibodies bind. So we go on a stepwise fashion and then just modify the virus as we go and engineer and modify the surface. So the top right, you see the gray virus, that's the original parent. And then at the bottom right, Mavis fondly used to call that clown vomit. But essentially, you have this whole modified surface of viruses that is now immunotransparent because the antibodies can't see those epitopes anymore because we engineered them, but it still delivers DNA very effectively. So our hope is we can use this to expand the number of patients that can be recruited to a trial, because a lot of us have seen have seen these viruses before, so have antibodies. And this is something that we're continuing to advance. But these are some of the steps that we're taking in order to continue to improve this platform. but I hope this gave you guys a 10,000-foot view of this virus that we've been able to hijack and modify this non-pathogenic virus for delivering genetic cargo that, as Andrew pointed out, can be useful in a gene therapy situation. I think I close with that, Andrew, for this session. So yeah, hand it back to you and let's tie it back to the big picture.
Well, thank you, Aravind. That was a tour de force of some of the incredible challenges and how your lab is really leading the way to surmount those challenges to bring gene therapy to patients. So thank you. In our closing last handful of slides, I wanted to share with you all just some ways that you all can stay on top of this field and maybe even get involved. And so, as Aravind mentioned, the American Society of Gene and Cell Therapy really has some great resources that you can access that you should really consider looking more into if this is something that's interesting to you. They have a whole website dedicated to educational materials. For example, all the way up to the left here, Gene Therapy 101, back to gene therapy basics, vectors, approaches, clinical trials and process, a great resource for you that would be accessible and something that we recommend. In addition, there's NORD, which I think is probably the coolest acronym around. It stands for the National Organization of Rare Diseases. It's basically an umbrella group for all these rare diseases that have typically someone's name attached with them. But NORD has a lot of information on gene therapy as well, because gene therapy is probably a major potential way to start treating some of these rare diseases like arrhythmic cardiomyopathy and like many of the channelopathies that we've talked about previously. And so rarediseases.org is a website where you can go check out NORD and some of their resources on gene therapy as well. And of course, I'd be remiss if I didn't mention the SADS Foundation. The SADS Foundation has tremendous resources, ways for you not only to learn about sudden death predisposing arrhythmia syndromes and AC, but also about gene therapy. I'll highlight they do have an e-newsletter. I subscribe to this as a provider. I love hearing about what they're doing, and you can as well. And finally, they are looking for subjects, individuals who may be interested in potentially learning more about gene therapy, in particular arrhythmic cardiomyopathy, ARVC here. And this is a portal that you can use if you have arrhythmic cardiomyopathy and potentially that PKP2 variant to get some more information and potentially be connected, at least through one potential gene therapy trial that's ongoing. Okay. And then finally, I'll close with just this last resource. This is a list of all the gene therapy trials that are ongoing right now, which you can access and check them out, see where they are in their development, and as new gene therapy trials get added, they'll be added to this resource. So another resource for you. So our take-home points from our time together. Gene therapy is the use of genetic material as a therapeutic. Arrhythmic cardiomyopathy, or AC, is caused by mutations in gene that make the cardiac desmosome. And when these mutations disrupt the desmosome, AC develops. Loss of function mutations in PKP2 are the most common cause of AC, and AC, and perhaps PKP2 specifically, may be amenable to gene therapy. And so with that, these are the people that I get to work with every day in my lab. Aravind knows some of these individuals, and so I'll just highlight their work and our funders. At this point, welcome any questions from you all. Thank you all for joining us.
Thank you very much, Dr. Landstrom and Dr. Asokan. We appreciate your information. And we have a question now from Tanya. Is gene therapy for everyone? I was diagnosed 26 years ago and beta blockers work. What about a defibrillator? Could I stop medicine with one?
That's a great question. I think gene therapy, like any therapy that's new, is something to consider, but may not be for everybody right away. I think if you have disease and you're on a beta blocker, you're feeling well, that beta blocker is preventing the arrhythmias from coming out. I think that's a really good start. Gene therapy holds tremendous promise down the road for being able to stop progression of disease and maybe even prevent disease. But I think when it first comes out, you're going to want to be very careful in how you utilize it in the clinic, and make sure that you're giving it to patients that really are struggling with the known therapies, the therapies that we know work, at least in the beginning. So I would say probably for the foreseeable future, that beta blocker is probably your best first line therapy. And hopefully down the road, gene therapy will play a role.
Thank you.
Of course.
Thank you. And we have another question from Jason. Could PKP2 scarring fatty tissue be reversed with gene therapy?
Well, that's certainly the dream. The trouble is, once scar sets in, it's really hard to make that scar go away and to get replaced by normal heart tissue. Probably where I think gene therapy has the most clinical utility is before you start getting that fibrosis, before you start getting that fatty replacement of the heart. Because scar is scar. If you think about, you cut your hand, and that cut turns into a scar, that scar is there forever. And really, it's really hard to kind of replace that without actually replacing the tissue itself. And so I would say the dream of gene therapy is hopefully to achieve that, but probably you want to try to initialize the therapy before there's significant scarring to get maximal effect.
Okay, Susan asks, do you see potential for gene therapy for DSP problems where data is switched versus missing? Or is PKP2 the most promising for gene therapy?
So PKP2 is probably the low-hanging fruit for right now, mainly because it's the most common gene that's mutated, the most common gene that's lost. And so the idea is that you can help the most people if you kind of go after that first. I do think DSP is a tempting target. I'm not aware of any gene therapy right now. Perhaps Dr. Asokan is for DSP specifically. But DSP is also a loss. And so in theory, even though there are single nucleotide variants that tend to cause DSP mutations, it's still kind of a loss. The protein isn't functional. And so replacing it might be a very reasonable way to fix the disease. It may be that you have to take a long QT approach where you suppress the misspelled one and replace with the good one. That may be, but DSP potentially could be amenable as well.
Anything to add there, Dr. Asokan?
No, Andrew covered it. I think it's the questions that the gene therapy space, or for that matter, any therapeutic modality space grapples with this access to patients and the prevalence. And I think at the moment, I'm not aware of any public information on DSP as a target being pursued. Yeah.
Okay, thank you. Jay asks, is PKP2 a good candidate for gene therapy because of the architecture or just the type of the mutation compared to the rest of the genes that are known to cause arrhythmogenic cardiomyopathy? Or is it just the first target due to the volume of people with it?
It's probably all those things together. And somebody may do a fancy business calculation and come to a different conclusion, but scientifically, the most people have mutations in PKP2. Typically, those who have mutations, the protein's not made, so it's relatively at least straightforward. And I know that something like this makes Dr. Asokan just really crazy because nothing is straightforward in this space. But it's easier just to replace a protein that's never made than, say, having a dysfunctional protein that's still floating around in the cell, still causing trouble, because then you have to make that go away and replace the protein. So PKP2 in theory is just a straight replacement, at least in theory. So I think combined with that and the ability to treat the most number of people, I think that would be the tempting target. Although, again, there may be other efforts underway and people have come to different conclusions.
Okay, thank you. Bill asks, can you explain how CRISPR comes into play with the technology you discussed?
Dr. Asokan, if you want to tackle that one, I can also jump in.
Sure, I can jump in and then feel free to chime in, Andrew. But yeah, so that's a good question. I think with CRISPR technology, the advantage is that we can very precisely target the regions that are mutated. And then the goal is you can replace those and put in the correct version or the copy of the DNA in there. The disadvantage is you now have to deliver the CRISPR machinery along with the pieces that need to be cut and paste in there. So you are adding to the complexity of the drug design process there. So we have to deal with not only the DNA that is being delivered, but also the CRISPR machinery that now has to be delivered to go and engage the target and work. The efficiencies of these will dictate, and the need on a per target basis will have to be, we have to match make those very carefully. To Andrew's earlier point, in this case, the gene for PKP2 fits into the virus and we can deliver the whole thing. And it's essentially a situation where there is insufficient levels of the protein. So you're restoring the levels of the protein. But there are situations where you might not be able to deliver the whole gene because of the size. And it's better to just go replace a small chunk of the gene, in which case CRISPR becomes a more interesting strategy there. So it's on a case-by-case basis that this is determined. Andrew?
I completely agree. I think of CRISPR as sort of like a scalpel, and it can be used to cut and change DNA at a very precise location. Right? The challenge is if you have a massive gene with lots of different spelling errors all through it potentially, where do you cut that gene precisely? And so one approach is to just, if you know that no matter where the misspelling is, the protein is down, you just replace the protein. I think and not take a CRISPR approach to do that, I think is a reasonable way to think about it. But there's lots of advances kind of in this area about how you can get really clever using CRISPR-Cas9. Dr. Asokan really leads the way here too, and I think there's going to be some situations where that therapy actually plays a clear role.
Okay, thank you both. Jason notes Johns Hopkins has discussed the potential for human clinical trials recently. Do you have any information or update on that effort for PKP2? And I know Crystal Tishnell is on the website webinar here. Oh,
yeah,
she actually commented here. Crystal wrote, we remain in conversations with companies to develop those initial trials, safety and efficacy first.
That's right.
Lots of challenges as discussed.
Well, I think Crystal's got a lot more information about this than I do. We certainly hope that these trials will be open soon. But as she indicates, and as we talked about last time, we have to do this the right way. This has to be safe. We don't want to do anything that puts anyone at any risk. We're in the business of curing disease, and so we have to do it the right way.
Exactly. Thank you, Crystal. Will this therapy be used for kids in the future? And when do you expect that as an option?
Yeah. So I am a pediatrician. That's my day job. And so I think about kids all the time. And kids, because kids can't say yes or no legally to therapies, we rightfully put them in this bucket of people that we call vulnerable individuals. And anytime you're dealing with a vulnerable individual or vulnerable population, I think the bar that we have to get over to show safety, to show that we have to treat them as kids and we can't treat them as adults when they are old enough to provide their approval and their permission, their consent. I think that bar is really, really high. And so for me, with AC, typically what I see is patients will have a mutation, but they won't develop disease until maybe early adulthood. And so in those situations, there may be a situation where you want to treat the child, but gosh, I'd love to be able to ask that child permission when they're an adult, to be able to sit down with them, not just their parents, but sit down with the adult and to go through all the potential risks and benefits of the therapy and have that conversation when they're old enough to make that decision. Will there be avenues for gene therapy for kids? Absolutely. But we have to do it the right way because of that vulnerable population.
Thank you very much. Are there any other questions? That's the last one in the chat. Well, we timed that perfectly. we're almost to the bottom of the half hour, so that's great. We thank you very much for your presentations. Very enlightening information, and we'll continue to monitor this and maybe have you back again. So thank you very much, everyone, and thank you to all of our families who joined us tonight. Have a good evening.